DNA microarray. DNA microarrays or nucleic acid arrays are a group of technologies in which specific DNA sequences are either deposited or synthesized in a 2D or sometimes 3-dimensional array on a surface in such a way that the DNA is covalently or non-covalently attached to the surface. So, we can analyze the expression of many genes in a single reaction quickly and in an efficient manner. DNA microarray technology has empowered the scientific community to understand the fundamental aspects behind the growth and development of life, as well as to explore the genetic causes of anomalies occurring in the functioning of human body. A typical microarray experiment involves the hybridization of an mRNA molecule to the DNA template from which it is originated. Many DNA samples are used to construct an array. The amount of mRNA bound to each site on the array indicates the expression level of the various genes. This number may run in thousands. All the data is collected and a profile is generated for gene expression in the cell. A microarray introduced in 1995 is a chip printed with thousands of variants of a particular polymer such as the cDNA, DNA or RNA. Each variant appears in a tiny dot, probe or spot containing many copies of the same sequence. The target sequences labeled with fluorescent tags are washed across the chip and the dots, when bound by the target sequences, fluoresce as shown in figure 1. The luminescent intensity of each probe indicates the relative quantity of the target present. A recent important application of microarrays is the tiling array, where nearly every sequence in a genome of at least some particular length, example 30 base pairs, is tiled on the array. Tiling arrays are powerful for genome-wide investigations and have many applications such as transcriptome mapping, protein binding sites mapping, etc. Various applications of microarrays end up with the same output format, that is, a high resolution image of thousands of variously illuminated little dots. A DNA array is used to probe a solution of a mixture of labeled nucleic acids and the binding of these targets to the probes on the array by hybridization, which is then used to measure the relative concentrations of nucleic acid species in solution. An array can be used to quantify an arbitrary large number of different nucleic acid sequences in solution. There are different methods of quantifying different nucleic acid sequences in a sample, including quantitative PCR, digital PCR and hybridization to optically tagged probe sequences.